Imagine stepping back to 1665, a time when, well, the very building blocks of life were just a complete mystery. Then, this remarkable scientist, Robert Hooke, he peered into this invisible world with a microscope he actually built himself. What he saw? It wasn't just small. It was like the first real clue in the mystery of life itself. These tiny, honeycomb-like sections in a thin slice of cork. And that observation, as simple as it sounds, it really set us on the path to understanding everything, from the tiniest bacterium right up to us. Welcome to the deep dive. Today, we're really going to focus and get to the absolute essence of life, the fundamental unit of life. If you're prepping for board exams or maybe competitive tests like UPSC or SSC, or even if you're just curious about how living things actually work, we're here to distill the most vital info you need from our sources. So let's jump right in. The big question. What are living organisms truly made of?
Well, in essence, they're made of cells. That's the term. These microscopic structures, they're the basic building blocks of, well, all living things. Understanding them is absolutely fundamental to biology, and it's a critical concept for your exams, for your whole academic journey, really. Robert Hooke's observation back in 1665, yeah, that was truly foundational. Like you said, he examined cork, saw these many little rooms or compartments. They reminded him of cells in a honeycomb. So he used the Latin word cella, which literally means a little room.
Ah, okay.
And this was profoundly significant because it was the very first time anyone had seen that living matter wasn't just one continuous blob, you could say. It showed it was made of these separate, discrete units. It completely shifted how we viewed life.
So Hooke really opened the door, didn't he? Who else kind of walked through it to help us truly understand the cell?
Oh, absolutely. The journey definitely didn't stop there. Just 9 years later, 1674, you have Antony van Leeuwenhoek. His microscopes were even better, and he looked upon water and saw something amazing. Free-living cells actually moving around.
Wow.
Yeah, so life wasn't just static structures, but these dynamic individual things. Then, fast forward a bit, 1831, Robert Brown identifies the nucleus inside the cell, a huge piece of the puzzle. In 1839, Purkinje gives a name to the stuff filling the cell protoplasm. So these individual discoveries, they started coming together into this really powerful idea, the cell theory. Matthias Schleiden, he was a botanist, proposed in 1838, okay, all plants are made of cells. Then just a year later, 1839, Theodor Schwann, a zoologist, says the same applies to animals. And crucially, he stated the cell is the basic unit of life.
Right, the basic unit.
Think of it like biology's unifying theory for life. It tells us, look, despite all the incredible diversity out there, a huge tree, a tiny bacterium, everything works on the same basic cellular plan. It's the bedrock, really. And then Rudolf Virchow added a critical piece in 1855. Omnis cellula is cellula.
Which means?
Basically, all cells arise from pre-existing cells. Life comes from life at the cellular level. And finally, much later, the electron microscope in 1940 that just blew everything wide open, letting us see the complex machinery inside the cell, the organelles, in incredible detail.
That's a huge shift in thinking. So, okay, cells are the basic units, but are all living things just like bags of these units? Mm-hmm. Or are there different ways they're put together?
That's a great question. There are different levels of organization. You have unicellular organisms. Here, a single cell is the entire organism. Think amoeba, chlamydomonas, paramecium, many bacteria. These are common examples you'll see.
Like one cell does everything.
Exactly. Then you have multicellular organisms. That's like most fungi, all plants, all animals. They're composed of many cells. These cells work together. They specialize, form tissues, organs, different body parts doing different jobs. But what's really amazing is that every single multicellular organism, even you and me, starts as just one cell. Right. That one cell divides and divides again, making more cells of its own kind. It perfectly illustrates Virchow's point cells come from pre-existing cells.
It really is mind-bending thinking we all start from one cell. But if everything's made of cells, are they all just identical little building blocks? I mean, a nerve cell is completely different from a blood cell, right?
You've hit on a really key point there. Cells are incredibly diverse, even though they're the fundamental unit. Yeah. Their shape and size, they're almost always directly related to the specific function they perform. So an amoeba, for instance, is constantly changing shape. That's how it moves and grabs food. But compare that to, say, a human nerve cell. It typically has a very fixed, specialized shape, long and branched, usually. Perfect for sending signals over distances. Our sources show this diversity even just within human cells. You've got sperm cells, bone cells, smooth muscle cells, blood cells, the ovum, nerve cells, fat cells. All look quite different because they do different things. And this idea of specialization, it goes even deeper inside the cell. Think about how a multicellular body has a division of Labor, heart pumps, stomach digests, right? Well, a single cell does too internally. It has specific components called cell organelles. Each organelle has its own special job. Some make new materials, others clean up waste. It's this intricate teamwork between organelles that lets the cell live and function.
Okay, we've got this internal division of labor. But what are the absolute must-haves, the core components every cell needs to, well, live and interact with its environment?
Good question. At a really fundamental level, almost every cell you look at, plant, animal, bacteria, shares three key features. You've got the plasma membrane, the nucleus, and the cytoplasm. These three together enable everything the cell does internally and how it deals with the outside world. Let's break those down because they're really critical for your understanding. Okay, first, the plasma membrane. You might also hear it called the cell membrane. This is the cell's outermost covering. Its main job is to separate what's inside the cell from the environment outside.
Like a boundary.
Exactly, but it's not just a passive wall. It's incredibly active. We call it a selectively permeable membrane.
Selectively permeable.
Meaning it carefully controls what comes in and what goes out. It lets essential things pass through, but blocks others. Very selective. Now, how things move across this membrane is vital. Some small molecules, like carbon dioxide or oxygen, they move by a process called diffusion. This is just the natural, spontaneous movement of a substance from where it's more concentrated to where it's less concentrated.
Okay, spreading out.
Right. So if CO2, which is waste, builds up inside the cell, it's more concentrated there, so it diffuses out. Oxygen diffuses in if the cell needs it and the concentration inside is lower. It's key for gas exchange. Water moves similarly, but its movement across this selective membrane has a special name: osmosis. Osmosis is basically the net diffusion of water across that membrane, moving from an area of higher water concentration to lower water concentration, or, put another way, towards a higher solute concentration.
Okay, let me see if I get this. Water essentially moves to where there's less water or more stuff dissolved, trying to balance things out. Is that right? And what happens to cells if you put them in different kinds of solutions? This feels like something that comes up in exams a lot.
Absolutely. It's a classic concept and very important. So imagine three scenarios. One, you put a cell in a hypotonic solution. Hypo means less solute, so the solution outside has a higher water concentration than the cell. It's very dilute. Water will move into the cell by osmosis, and the cell will swell up. You can see this if you put a deshelled egg or some dried raisins in plain water, they get plump. Two, an isotonic solution. Iso means same. The water concentration outside is the same as inside, so there's no net movement of water, and the cell stays the same size.
Right.
Three, a hypertonic solution. Hyper means more solute, so the solution outside has a lower water concentration than the cell. It's concentrated. In this case, water moves out of the cell by osmosis, and the cell shrinks. Like putting that deshelled egg or raisins in concentrated salt or sugar water, they shrivel up.
Okay, hypotonic swells, isotonic stays same, hypertonic shrinks.
Got it. Exactly. And this principle is fundamental for things like how plant roots absorb water. Oh, and the plasma membrane itself. It's flexible, made of lipids and proteins. This flexibility lets cells do things like endocytosis. That's how an amoeba actually engulfs its food. Now, for plant cells, and also fungi and bacteria, there's another layer outside the plasma membrane, a really important one, the cell wall. This is a rigid outer cover, usually made mainly of cellulose in plants. It provides structural strength.
So it's tougher than the membrane.
Much tougher, yeah. And because it's rigid, it leads to an interesting phenomenon called plasmolysis. This is another key term for exams. Plasmolysis happens when a living plant cell loses water in a hypertonic solution. The inside part, the protoplast, shrinks and pulls away from that rigid cell wall. You can actually see this under a microscope, maybe using a rheoliphin salt solution. But the big advantage of the cell wall is that it lets plant cells and fungal bacterial cells survive in very dilute hypotonic environments without bursting. When water rushes in, the cell contents swell and push against the wall pushes back, creating pressure, and that stops the cell from exploding. Animal cells, lacking this wall, would just burst in pure water.
All right.
So moving inwards from the boundary layers, we get to the nucleus. The cell's command center, essentially. You might remember seeing it in diagrams or maybe in lab preps like onion peels or cheek cells. It's that dark staining, usually spherical or oval structure, often near the middle. It's wrapped in a double layer called the nuclear membrane, which has tiny pores. These pores control what goes in and out between the nucleus and the main cell fluid, the cytoplasm. Inside the nucleus, you find the chromosomes. Now, these are usually only clearly visible as distinct rod shapes when the cell is actually dividing. Chromosomes are incredibly important. They're made of DNA, deoxyribonucleic acid, and proteins. DNA is the molecule that carries all the hereditary information passed down through generations. Specific functional segments of DNA are called genes.
Right, genes are on the DNA.
Exactly. When the cell isn't dividing, the DNA looks more like a tangled mass of threads that's called chromatin material. It only condenses into those neat chromosomes for cell division. So the nucleus has a massive role. It controls cellular reproduction, cell division, and it directs all the cell's chemical activities, basically determining what the cell becomes and how it functions.
That's a really clear picture of the nucleus as this control hub. But does every single living cell have this kind of well-defined nucleus with a membrane around it? Or are there variations?
That's a very sharp question, and it leads us straight to a major division in the living world: prokaryotic versus eukaryotic cells. In some organisms, mainly bacteria, the nuclear region isn't enclosed by a membrane, it's just sort of there in the cytoplasm. This undefined nuclear region, which just contains the nucleic acids, is called a nucleoid.
Nucleoid, okay.
Organisms with cells like this lacking a true membrane-bound nucleus and also lacking most other membrane-bound organelles are called prokaryotes. Pro means primitive or before. Their internal organization is much simpler. Many of their functions just happen in less organized parts of the cytoplasm. Now contrast that with eukaryotic cells. Eu means true. These cells do have a proper nuclear membrane enclosing the nucleus. They also have a whole suite of other complex membrane-bound organelles within their cytoplasm, which prokaryotes mostly lack.
So eukaryotes are more complex internally.
Much more complex. And those internal membranes are incredibly significant. They create compartments, allowing different chemical reactions to happen simultaneously without interfering with each other. It allows for much greater efficiency and specialization. This membrane difference is so fundamental. Even viruses, which aren't really cells and lack membranes entirely, they can only show signs of life inside a living host cell, hijacking its machinery. Okay, so let's explore those fascinating membrane-bound structures inside eukaryotic cells of the organelles. These are what really give eukaryotes their complexity. First up, there's the endoplasmic reticulum, or ER for short. It's this huge network of membranes forming tubes and sheets throughout the cytoplasm. Think long tubules or roundish sacs called vesicles. Its membrane is actually quite similar to the plasma membrane.
And it does, wait.
Well, there are two types, and they do different things. There's the rough ER, or RER. It looks rough because it's studded with tiny dots called ribosomes. Ribosomes are protein factories, so the RER is where many proteins are made, and then these proteins are sent off to where they're needed. Then there's the smooth ER, or SER. No ribosomes, so it looks smooth. Its main job is making lipids fats, which are also vital for the cell.
Lipids like for the cell membrane.
Exactly. Some of those proteins from the RER and lipids from the SER are used to build more cell membrane. That process is called membrane biogenesis. The cell makes its own skin, essentially. The SER has other jobs too, like detoxifying poisons and drugs. This is a really big function in liver cells, for example. So overall, the ER acts like a transport network, especially for proteins, and a surface for lots of biochemical reactions. All right, next we have the Golgi Apparatus, sometimes called the Golgi complex or Golgi body, named after Camillo Golgi, who first described it. Structurally, it looks like a stack of flattened membrane-bound sacs called cisterns, kind of like pancakes stacked up, often near the ER.
Okay, like a stack of pancakes. What's its function?
Think of it as the cell's post office or finishing and packaging center. It takes products made in the ER, modifies them, sorts them, packages them into vesicles, and then dispatches them to various destinations inside or outside the cell. It can also make complex sugars from simple ones, and importantly, it's involved in forming lysosomes. Which brings us to lysosomes. These are interesting little sacs. They're membrane-bound and filled with really powerful digestive enzymes. These enzymes are actually made by the RER and then processed by the Golgi.
Digestive enzymes, so they break things down.
Precisely. They're the cell's waste disposal and recycling system. They break down foreign material that gets into the cell, like bacteria or old, worn-out organelles, keeping the cell clean. They can digest complex molecules into simpler ones, but they also have this nickname, suicide bags.
Suicide bags, why?
Well, if the cell gets seriously damaged or stressed, the lysosomes can actually burst open, releasing all those digestive enzymes into the cell's cytoplasm. This effectively digests the cell from the inside out. It's a drastic measure, but sometimes necessary.
Wow, so they're the cleanup crew, the recyclers, and the self-destruct button if needed. That's quite a responsibility.
It really is. Okay, moving on to the mitochondria. These are very famous, often called the powerhouses of the cell.
Right, I've heard that. Why powerhouses?
Because they are the primary sites of cellular respiration. They take fuel molecules, like glucose, and break them down to release energy in a form the cell can actually use. That usable energy form is a molecule called ATP adenosine triphosphate. ATP is like the energy currency of the cell. All the cell's activities that require energy spend ATP.
Okay, so they generate the cell's power supply.
Exactly. Structurally, they have two membranes, an outer one that's fairly smooth and porous, and an inner one that's folded up dramatically into cristae. These folds vastly increase the surface area inside, providing more space for the chemical reactions that generate ATP. And here's something really unique about mitochondria. They have their own DNA and their own ribosomes.
Wait, separate DNA from the nucleus?
Yes, it allows them to make some of their own proteins independent of the nucleus. It's a fascinating feature, possibly linked to their evolutionary origins. Now, another organelle type, but this one's found only in plant cells, the plastids. There are two main categories: chromoplasts, which are colored, and leucoplasts, which are white or colorless. The most important chromoplasts are the chloroplasts. These contain the green pigment chlorophyll, which is absolutely essential for photosynthesis, the process plants use to make food using sunlight.
So chloroplasts are for photosynthesis.
Correct. Chloroplasts might also contain other pigments, like yellows and oranges. Leucoplasts, being colorless, are mainly for storage. They store things like starch, oils, or protein granules. And just like mitochondria, plastids also have their own DNA and ribosomes. Finally, let's talk about vacuoles. These are basically membrane-bound sacs used for storage. Could be solid or liquid contents. Now, their size differs a lot between animal and plant cells. In animal cells, if they're present, they're usually small and temporary. But in mature plant cells, there's often one huge central vacuole that can take up, 50 to 90% of the entire cell volume.
Wow, that's massive. What does it do?
It's filled with cell sap water, sugars, salts, amino acids, proteins, waste products. This big, full vacuole pushes outwards against the cell wall, creating pressure called turgor pressure. This pressure provides rigidity and support to the plant cell, helping keep plants firm. It's also a major storage site. In some single-celled organisms like amoeba, you find specialized vacuoles. Food vacuoles hold ingested food, and contractile vacuoles pump out excess water to maintain balance.
Okay, we've gone through this incredible list of components, this complex machinery inside the cell. Now, how do these cells actually multiply? How do we get more cells for growth or repair or even reproduction?
That's the next crucial step: understanding cell division. New cells have to be formed for an organism to grow, to replace old or damaged cells, and, importantly, to form the specialized cells needed for reproduction, the gametes. The process that makes new cells is called cell division, and there are two fundamental types you need to know, especially for your exams: mitosis and meiosis. First, mitosis. This is the type of cell division that most of the cells in your body use most of the time. Its main purpose is for growth, making more cells so an organism gets bigger, and for repair, replacing cells that are worn out, damaged, or dead.
Okay, growth and repair.
Exactly. In mitosis, one cell, we call it the mother cell, divides to produce two daughter cells. And the absolutely key thing about mitosis is that the two daughter cells are genetically identical to the mother cell. They have the exact same number of chromosomes. This ensures consistency, making sure all the body cells mostly have the same genetic blueprint. Now, the second type is meiosis. This is a more specialized process. It only happens in specific cells located in the reproductive organs, tests and ovaries in animals, or specific parts of flowers and plants.
Only for reproduction, then?
Primarily, yes. Its purpose is to produce gametes, sperm and egg cells, or pollen and ovules. These are the cells that will fuse during fertilization to create a new organism. Meiosis is different from mitosis in a couple of key ways. First, it involves two rounds of division, one after the other. So one starting cell produces 4 new cells, not just 2.
4 cells instead of 2.
And here's the most critical difference. These 4 new cells produced by meiosis have only half the number of chromosomes as the original mother cell.
Half the chromosomes. Wow.
That reduction is essential for sexual reproduction. Think about it. If sperm and egg each had the full set of chromosomes, when they fused, the offspring would have doubled the normal number. Meiosis halves the number in the gametes, so when they fuse, the correct full number is restored in the offspring. So just to recap the big picture, the cell really is the fundamental unit. Structurally and functionally, it's the basis of all life. Its amazing abilities come from this specific organization, the membrane controlling passage, the nucleus directing operations, and all those specialized organelles carrying out specific tasks within the cytoplasm. We've touched on its discovery, unicellular versus multicellular forms, the core structure, the jobs of the Golgi, lysosomes, mitochondria, plastids, vacuoles, and finally how cells divide. Mitosis for growth and repair, keeping the chromosome number the same, and meiosis for reproduction, having the chromosome number to make gametes.
It is truly astonishing when you think about it. All that complexity, that self-sufficiency, packed into something usually microscopic. It definitely makes you wonder about the bigger questions, doesn't it? About how life is defined, how it continues, and just the incredible balance maintained inside every living thing, starting at this cellular level, from Hooke's little rooms to this intricate universe inside. We really hope this deep dive into the cell has helped clarify just how vital and complex this fundamental unit is for all your studies. Stay tuned for more exam-ready insights, and remember to subscribe to never miss a topic breakdown on the deep dive. Thanks for joining us. In this deep dive, we cover the following topics: the discovery of the cell, unicellular and multicellular organisms, the structural organization of the cell, including the plasma membrane, cell wall, nucleus, and cytoplasm, detailed functions of key cell organelles, the Golgi Apparatus, lysosomes, mitochondria, plastids, and vacuoles, and finally, cell division, mitosis and meiosis,